osmosis. So this is part of the 3.2.3 transport across cell membranes topic. So osmosis explained in terms of water potential. And there is a required practical associated with this, which is production of a dilution series of a solute to produce a calibration curve with which to identify the water potential of plant tissue. So first of all, it's important to know that cell membranes are partially permeable. So that means they let water or very small molecules through, but not starch, which is large. And the definition of osmosis is the passage of water from a region of where it is a high water potential to a region where it has a lower water potential through a partially permeable membrane. Key things about osmosis, again, it's still going down a concentration gradient from high to low, so it does not require energy, and it's the movement of water. So solutions and water potential. So water potential, which the symbol is this, is measured in kilopascals. And under standard temperature and pressure, the water potential of pure water is 0. So everything else is negative in comparison. So pure water is 0. As you start adding solutes, it becomes negative. So the addition of a solute to pure water will lower the water potential. Therefore, the water potential of a solution must be less than 0, or it must be negative, unless it's pure water. And the more concentrated a solution, the lower the water potential and the more negative it is. Water will move from a less negative, for example minus 20, to a more negative, for example minus 30, water potential. So it goes from a less negative to a more negative water potential. And you can find the water potential of a cell itself by placing it in a series of dilutions of a solute solution, such as a sugar solution, and then where the place that there's no net gain or loss must be the water potential of the cell, because it's equal to the water potential of the solution if there's no net movement. Weak solutions or dilute solutions have a high water potential, closer to 0 or less negative, whereas concentrated solutions have a lower water potential or more negative. But when the water potentials are equal, so it's an isotonic solution, there is a dynamic equilibrium, so there's no net movement of water. As I said, water moves from a high water potential to a low water potential, and the highest possible water potential you can have is 0. So because it goes from a less negative to a more negative, it would go from minus 200 to minus 400 kilopascals and minus 400 to minus 600 kilopascals. It always goes from a less negative to a more negative water potential. So now we want to look at osmosis in animal cells and how that differs from osmosis in plant cells. So animal cells, for instance a red blood cell, doesn't have a cell wall, and this means that it's very vulnerable to changes of water potential. So animal cells, like red blood cells, contain some solutes in their cytoplasm. So if you placed a red blood cell in pure water, it's going to absorb water by osmosis, because the cell itself has a lower water potential or a more negative water potential. And as water moves in, because the cell membranes of red blood cells are thin, it will actually rupture and burst the cell, which is called hemolysis or hemolysis. Lysis means breaking open the cell. Hemo is to say that it's like red blood cells. So red blood cell hemolysis or hemolysis. So this is shown here. If you put in a hypotonic solution, which means it's a dilute solution, what's going to happen is the water's going to move in and it's going to rupture the cell membranes and it's going to burst. On the other hand, if you put it in a concentrated solution, a hypertonic solution, what will happen is the water will move out of the red blood cell by osmosis and it will shrivel up. So it's really important in our blood that our blood is isotonic. Isotonic means that our blood plasma has the same water potential as the red blood cell. Because if there's the same water potential outside and inside, there's going to be no net movement of water. So here is what we see, that in a dilute solution, in like a pure water solution where it's hypotonic, there is a higher water potential outside and a lower water potential inside the cell. Therefore, osmosis happens and water moves in, and in an animal cell, the cell will swell and burst and die. Lysis, which is rupturing. If it's in an isotonic solution where there's an equal water potential inside and outside, there's going to be no net movement of water, so there's going to be no change. If it's in a concentrated solution known as hypertonic solution, there's going to be a low water potential outside and a higher water potential inside. Therefore, because it goes from high to low, or less negative to more negative, water's going to move out by osmosis and the cell is going to shrink and shrivel. Now, plant cells have a different response because of the fact they have a cell wall. So a plant cell that is placed in a less concentrated solution, a hypotonic solution, so like a dilute watery solution, what will happen is water will move in by osmosis and the cell will expand, the vacuole will expand, and the contents will push on the cell wall. But the cell wall, because it's quite strong, because it's made of cellulose, will push back and resist the pressure of the vacuole expanding, and instead the cell will become turgid. Eventually the water potential outside will equal the water potential inside and there'll be no further net movement of water and the equilibrium is reached, so it'll just stay turgid. On the other hand, if you put a plant cell in a solution that is more concentrated than the cell solution, loses water by osmosis. So water will move out by osmosis, the cell will become flaccid, the plasma membrane starts to become pulled away from the cell wall, and at this point, when the cell membrane comes away from the cell wall, the plant cell is said to be plasmolysed. And now what happens is the external solution actually fills the gap between the cell membrane and the cell wall. And eventually some parts of the membrane become torn away or pulled away from the cell wall, and the cell dies at this point. So some practice exam questions now. Give two similarities in the movement of substances by diffusion and by osmosis. So they're both going down a concentration gradient from a high to a low concentration, and they are both passive processes. They do not require energy from respiration. Another one, Tradescantia is a house plant. There are small hairs on its flowers. These hairs are made of cells. Figure 1 shows the appearance of cells from one of these hairs after 20 minutes in distilled water. Figure 2 shows cells from another hair after 20 minutes in a solution of potassium nitrate. What does figure 2 suggest about the permeability of the plasma membrane surrounding these cells? That usually it is partially permeable and it allows water to pass through, but not potassium nitrate. What is present in the space labelled F? Explain your answer. Well, when it starts to become plasmolysed, the external solution starts to fill the space, so it will be potassium nitrate because the cell wall is permeable. It's just the cell membrane that's only partially permeable. How would the water potential of the sap in vacuole of cell E differ from the water potential of the sap in the vacuole of cell D? Explain your answer. So the water potential is more negative or lower in E, therefore water is removed. There's greater solute or sap concentration in that cell.